computer. We'll start. Okay. Welcome this morning to our Centers of Excellence in Iron and Heme Disorders, interviews with giants in iron. Today, we're privileged to hear from Dr. Tom Ganz, who is a distinguished professor in the Department of Laboratory Medicine and Pathology at the University of University of California, Los Angeles. Thank you so much, Tom, for agreeing to be interviewed today and help us to essentially immortalize memories and stories of people who led the field and brought us through our iron. I can't think of the right word right now, but the iron renaissance, I guess is what I want to say. How's that?
That's
How's that?
good. I
Yeah.
like that.
Yeah, I like it too. And I am not going to take a lot of time on an introduction for Tom because we want to hear this story more. And so I'm going to turn the time over to Cade Leverage, who is our MD PhD student who will be conducting the interview. And I'll probably chime in periodically when I want to ask a question. So thank you so much. I'm going to stop sharing and it'll just be the three of us on this recording. Okay, Cade.
Awesome. Well, I am very much looking forward to this interview. And I just wanted to start asking, could you just give us a background of you and your family?
So I was born in 1948, which was 3 years after the end of the Second World War, in Prague, Czechoslovakia. I was born into a secular Jewish family. My father was a biomedical scientist, a cardiologist who was effectively an equivalent of today's MD PhD. And he was interested in coronary circulation and in heart function and in how to measure heart function. He developed interesting treatments for heart disease. And he was actually quite a pioneer in the cardiovascular field. My mother was executive secretary, essentially. She was a woman of many skills who was denied education because of the war circumstances and who tried all her life to learn things on her own because she felt the gap of not having been educated up to her potential. I have a brother, Peter, who is a cardiologist at UCSF, University of California, San Francisco, also in research. And we lived in Prague until I was 18 years old. And I had I would say, a happy childhood, despite the political circumstances and the oppression of the communist regime. I didn't feel it the same way as my parents did. I had my friends. I interacted with a nice group of people. I was socially active. I did some sports. I was interested in science. And at the age of about 15, when I was still in elementary school, people came by looking for talented mathematicians, physicists, people like that, people who had the potential to develop into scientists. And they picked me out, and I went to a special high school, which is kind of experimental. It's kind of a response to the Sputnik era. And I had remarkably 10 hours of math a week, 5 hours of physics a week, computers when nobody knew what computers were. I learned programming, and I had a very high level, very elite education, essentially. At the age of 18, I illegally emigrated to the United States, illegally from the point of view of the Czechoslovak government, who promptly sentenced me to 2 years in jail. And my parents, my brother and I came to Los Angeles, and I started at UCLA. And they looked at my record and they enrolled me as a sophomore. So I skipped my freshman year and took a lot of physics classes, math, no biology. I thought biology was way too soft. I thought biology consisted of naming the parts of flowers, which didn't really interest me. And so I embarked on essentially a scientific basic science, physics, math education. Went to UCLA, took one biology class, which I believe was evolution for poets or something like that. And because I had to, it was required. And ended up at Caltech for a in a PhD program for applied physics. And really enjoyed it, worked on superconducting circuits, which was a novelty at that time. And really enjoyed doing experiments with my hands, constructing equipment, understanding superconductivity and what can be done with it in practical terms. Then this all was happening in the background. There was the Vietnam War. And what happened is that, unbeknownst to me, the area of applied physics that I was working in was really funded by the Navy, the military. And when the budget started, when the war started winding down and the budget started getting smaller, one of the first things that got cut out was my area of science, superconducting circuits. And so I started looking for something else to do. And I heard that interesting things were happening in biology. And so I started paying attention. I took a course from a professor named Max Delbruck, who was a Nobel Prize-winning phage biologist. And he was a physicist originally, so he was the kind of guy who spoke to me in terms I could understand. So his classes discussed the biophysics of neural transmission, channels, ionic channels, things of that sort. And it was all very interesting. And then I took another biology class, which was more biochemically cell biology oriented, from Lee Hood. And that was a lot of fun. So that opened my eyes that something interesting was happening in biology. And I decided I was going to become a molecular biologist. So I took the courses, started doing some work in the lab, and then I got my PhD in applied physics, and they told me, well, if you want to continue getting educated in biology, you really can't do that. We only give one PhD, so you have to do it on your own. And I said to myself, well, maybe I can do it. I'll just go to medical school. And so I went to medical school to become a biomedical scientist. So in 1974, which was 2 years before my PhD, I enrolled in medical school at UCLA. So I was simultaneously at Caltech and at UCLA. And I finished my PhD and finished my MD 2 years later. So I'll pause in case you would like to ask any questions or clarifications.
Yeah,
How did you do that?
Yeah, I think that's the first time I've ever heard of somebody doing their PhD and MD at the same time like that. It's pretty amazing.
This was before the MD PhD programs that kind of standardized this routine.
Yeah.
And I just somehow did it on my own because I was curious. I was not a very practical person. So, the people amaze me today. They have career planning. I didn't know what the career planning was. I had no idea what I was going to do. I just wanted to do something interesting and get paid for it.
Yeah.
That was my career planning. And yeah.
That's awesome. I'm trying
cool.
to think about Pasadena over to UCLA, the commute.
It wasn't that bad.
Yeah, back then probably
And
not.
at that time, the traffic wasn't quite as bad as it is now. And also, at Caltech, I could start at any time I wanted to. So my usual time was 11 to 8 or 11 to 9. So I skipped the traffic altogether. I remember rolling out on the Pasadena freeway and waking up at the same time around 10:30 or so, or 11, and then working until I got tired and I went home for dinner.
There we go. That's amazing, and that you could absorb all those materials and be performing the experiments necessary for your PhD at the same time. I always advise students, have always advised students, it takes a while to get your systems up and running, all the tools you need to be successful to answer the questions you were trying to answer. But when you get them, bring in a cot because
Yes.
just run with it.
And that's what happened to me effectively. I sort of prepared everything in my first 2, 3 years at Caltech, got everything set up to where I could just get data. And at that point, I got help from another graduate student who was going to take my project over when I'm done with it. And so I didn't have to, I wasn't alone anymore. So somebody was working with me and we finished the project and he took it over.
Lucky you and lucky him.
Yeah, he got a running start,
Yeah.
and I got to occasionally read the textbooks for medical school.
So
Well,
Yeah,
fun.
I think I would love to hear about, you've done med school, you've done your PhD in applied physics. Tell us about how you got involved in iron metabolism and kind of where that took you throughout your career.
yeah. So what happened is that let me just turn the phone off.
Oh, thank
I'll
you.
just send a message that I am not available. I am not. Okay. So what happened is that I went to medical school. I decided to do internship and residency in internal medicine. I did that. And then the next step is a fellowship. And for the fellowship, I did the kind of the minimal clinical program and went back to the lab. And my first laboratory experience was with a cell biologist named Harvey Hirschman. And I worked on an interesting new idea. At that time, today it seems trivial, but it was the question about how metals regulated the transcription of metallothionein. So I worked on my first iron-related project was not really iron-related because the metals I used were zinc and cadmium. And so zinc and cadmium, if you take HeLa cells or any other cell line probably, and you treat them with high concentrations, so zinc or cadmium, they will massively induce the transcription of metallothionein genes. And that was used as a model of transcriptional regulation. At that time, we didn't quite understand transcriptional regulation the way we do today. So the model promoter was the metallothionein promoter. And I did a little bit of work. It was effectively for me a postdoc because I had gotten my PhD in applied physics. So I had to relearn all the biology that happened and all the experimental technology that happened between the time that I finished at Caltech, went to medical school, and was never in really full-time in a biology lab. So I remember my first day in the lab, I looked at the pipette man, and they were pipette men like we have today already at that time. But they were unfamiliar to me because when I left the lab at Caltech, they were fixed pipette men. They were marked 20, 50, 100, 10, and you couldn't adjust them. They were just fixed. And so I came to the lab and I didn't took me a while to figure out these guys are adjustable. You can actually set the volume. And I didn't know what those numbers were there for. And eventually I asked somebody and figured it out. And a technique that seems funny today, one of the techniques we used uh, was Sanger sequencing at that time. And, the day that I got 100 bases was a good day.
Yeah.
And we would do the sequencing reactions. We would load them on a gel. There were huge gel apparati that huge, and the bigger, the better the resolution. And and then the gels themselves were like a millimeter thick, and you had to essentially put them on a solid support to be able to handle them. And then you would do autoradiography. You would take the gel and you put a huge piece of film on it, and next day you could read the sequences. And so I did a bunch of sequencing in the rat methylthionine promoter. That was our model. And it was a co-author in a paper. And then I decided I learned enough in that lab. I was going to do something that was more medically immediately related. And I talked to my wife at that time, and she said to me, I know this really crazy smart guy that you would hit it off with. That was my introduction to a person who ended up my mentor. And he is a hematologist who studied neutrophils, human neutrophils, and in animal models, neutrophils. And what puzzled him is that at that time, the idea was that neutrophils use hydrogen peroxide and superoxide to kill bacteria. And that was the dominant paradigm. And he started doing experiments with cells from people who did not make hydrogen peroxide, had the disease called chronic granulomatous disease. And he noticed that they killed just as well as the wild type. There was only a small number of bacteria that were preferentially killed by wild type compared to CGD cells. Most bacteria were killed equally well by both. And so the question was, what was killing the bacteria? So I came to his lab, and my task was he wasn't interested in neutrophils at that time because he had already done work on neutrophils. He decided that neutrophils in humans did not have this killing material that he was looking for. And so he told me to look at monocytes. And I ground up monocytes, put the extracts of bacterial plate, fractionated the extracts. I would, sometimes get unbelievable, visible amounts of monocytes that with 10 ml, 30 ml of monocytes, grind them up, fractionate them, look for the killing material. And it was pretty hopeless, actually, at the beginning. I wasn't getting anything. One day, we let the column run too long. This was a size fractionation column, polyacromide gel-based column. We just sort of forgot it was running, let it run too long, and got a huge peak after the void volume. So a huge peak at the end of so when the salt was supposed to come out, we got this huge peak after that. After the smallest molecules came out, we got a huge peak. So we were trying to figure out what happened. Later on, we figured out what is that the material that we were looking for had a specific affinity for the polyacromide matrix. So we were doing affinity purification. And so the material came out after salt. And we had a very interesting group of peptides, which we named defensins. And the next surprise was they were not coming from monocytes, but from neutrophils. They had been missed on the original screen because of their special properties, because they bound to the column and came out when after the runs stopped. They were still on the column. And so my first paper, significant paper, was a description of this group of peptides in human neutrophils, which we named defensins, and their role in host defense. And I spent after that the next from 1985 maybe to 2000, 15 years working on defensins, and they turn out to be a huge family of genes. And they had all kinds of roles in host defense, and we characterized the genomic locus. It's a huge family, and I was not happy. At the beginning, I was happy because I had some new stuff, but I was not happy because this family had some properties that made it nearly impossible at that time to study. And the reason that it was difficult to study is there was a highly redundant peptide family where the genes have just proliferated. They were different in mice than in humans. Every animal had their own evolutionary history. You couldn't knock them out because it was like megabases of genes that were highly repetitive, highly polymorphic. So later on, I found out when I was studied the polymorphisms that my friend had 13 copies of a gene of which I had none. And so it just was a mess from my point of view. There's still people studying that family today. It was a mess. I was looking for something else and to do. And one day, when I was studying urinary defensins, I noticed a peak in the chromatogram that actually we had not seen before, and the material didn't react with the anti-defensin antibody. So at that time, we were studying urinary defensins, which are secreted by the mucosa of the urinary tract, urogenital tract. And these are host defense peptides that kill bacteria. But one peak did not react with the antibody that we had at that time that we used to characterize this group of proteins. And we just got some basic information on it. We sequenced the peptide. We found the gene, and in the at that time nascent Human Genome Project, we found the gene. It hadn't been even noticed because it's such a small gene. These peptides are encoded by very small genes, so it didn't pass the filter at that time. And so we pretty soon had the sequence. We had the genes involved. The gene involved. And I looked at that and I said to myself, this really isn't enough. I have no clue what this new peptide does. We knew it was made in the liver, and we knew it was mildly microbicidal, so we named it hepcidin. This was in the 1990s. And we put it in GenBank. Around 1998, we put it in GenBank, which was at that time the reference database. And I was at a meeting on antimicrobial peptides in France when this guy walks up to me. I didn't know him. Pierre Brissot walks up to me and says, that peptide you put in the database, we found that gene on subtractive hybridization between mice that were iron overloaded by giving them injections of iron, and mice that were the reference mice, which were on normal iron diet. And this peptide, the gene encoding it, is highly overexpressed in the iron-dosed mice. And I looked at him, and I didn't know what to do with the information. I had no idea what iron biology was about. I worked in a completely different field. I went home and I discussed it with the people in the lab. And my technician at that time, Erica Valor, says to me, we've got to do something with this stuff. I spent a year doing all this, and we can't just abandon it. We need to do something with it. And I said, okay. Sooner or later, this guy we will establish a working relationship with Brissot, and when he finishes the mouse project looking at this peptide, we will co-publish with him, and we will describe what we found in humans. They will describe what they found in mice, and we'll co-publish in JBC. So in 2001 or maybe 2000, maybe we send in parallel manuscripts describing hepcidin. There was one more thing, which is that I noticed that hepcidins have 4-disulfide bonds, and that there was a Drosophila peptide that kind of looked like that was inducible by LPS in Drosophila. And so I told Brissot at that time that they should inject their mice with LPS and see if this peptide is inducible with LPS. And they did that experiment, and that was included in their paper in JBC. So we had a peptide that was induced by iron, induced by LPS. And so this was beginning to look interesting. And what made me very happy is it wasn't a part of a large peptide family, which means that we could knock it out and see what happens. And so we were making preparations to knock it out when a team in France announced that they had accidentally knocked out hepcidin by knocking out a neighboring gene, the USF2 gene. They had studied USF2, which is a transcription factor, and they studied its effect in diabetes. And one of the people in the lab had come from Brissot's lab, and he knew the hepcidin story. And he says, USF2 is right next to hepcidin, and we should check if we knocked out hepcidin when we knocked out USF2. And sure enough, they had knocked out hepcidin, and the mouse had a black pancreas, very dark-looking pancreas, and was massively iron overloaded. So that was the first work that described the function of hepcidin, was this accidental knockout of hepcidin in mice. And it was complicated because unlike humans, the mice have 2 hepcidins. You HAMP1 and HAMP2. And it was a bit complicated, and the relationship to USF2 was not well understood. And in the end, though, what emerged from this, when we thought about it and other people thought about it, was that most likely hepcidin was the iron regulatory peptide that people had been looking for. And so I completely turned the lab around, and I said to them, look, we're going to work on iron. This is it. I'm done with antimicrobial peptides. And that's what happened. So that was the most exciting, I would say, period in my life at that time, because suddenly there was something that really looked interesting, novel, tractable. I could use the tools of modern biology to attack the problem and figure out how this works.
I remember it well. It was such a great time in the iron field. Just right and left things being discovered that explained human diseases. And it was just really.
It was crazy because I knew nothing about iron. So in the morning, I would go to the textbooks to try to figure out. Fortunately, I was so ignorant that I didn't just know anything about iron. I didn't know the bad stuff that was wrong either. So I was completely unprejudiced, and ignorant is another word for unprejudiced. But I would sort of go back to the articles, reread them, go back to the lab, try to figure out how things work, and then go back to the articles and read them again to try to understand how I can fit the new data to the old story. And it was very exciting. I barely slept. It was just one thing after another. Whatever we touched ended up being interesting. And that's kind of the beginning of a field. That's how the beginning of any field really is, because you have to discover everything. And at the same time, whatever you touch is new.
Yeah.
So it was, as Diane described it, it was that kind of time.
Yeah. I actually distinctly remember you sharing hepcidin to us. Um, and we had ferroportin with a GFP moiety on it. And I put the hepcidin on the cells and looked under the scope and started yelling down the hallway.
Yep.
I was like
Yep.
jumping
And
around going, it's the ligand for this. It
yes,
causes it to be downregulated. Anyway, I remember
to,
the
to be
emotion
to
of it.
be honest with you, I did not think of that kind of a, I did not think that you could actually look at the receptor just by looking at its endocytic property. Because to my mind, the endocytosis was kind of the last step when you overwhelm a receptor and that it gets sort of down-regulated by the ligand. That was, in my mind, the signaling pathways that precede that were the interesting thing. But you guys were in a different mindset. You were looking at endocytosis.
Correct.
Yeah, and I sort of, it took me a few days to wrap my head around that, the endocytic effect, and that you could use that to show how hepcidin actually works. Uh,
I just
so
remember the excitement of it. It was
yeah,
just,
I do
I could
too. I do
hardly
too
sleep
because
too.
because shortly thereafter you sent us the cell line and we saw it ourselves.
Yeah.
Um, it took us a while to get the conditions right, but yes, the endocytosis was the critical ingredient.
Yeah.
And
And then you guys showed the binding. I mean,
and then we showed the binding because
yeah.
what we could do, well, we could do several things that at that time nobody could do. One of the things was we could make synthetic hepcidin. Second is we looked at the hepcidin sequences and we noticed that we it's a peptide that's very hard to radio label to high intensity. So what we did is we switched one of the amino acids for the zebrafish amino acid that was in that position, and that happened to be a tyrosine, and we could iodinate the tyrosine, radioiodinate the tyrosine, and that synthetic peptide was so hot that we could actually quite easily show the endocytosis and the binding and the displacement from the receptor by cold hepcidin. So we could do a whole bunch of things. And the other interesting story is how we got our first milligram of hepcidin. I think I told the story many times, but I'll tell it again, which is that I still had some leftovers from the antimicrobial peptide life. So I went to Sweden for a meeting, and in the airplane, I went to the bathroom and I noticed that I had a little bit of burning on urination and some urinary symptoms. And in the typical medical fashion, I ignored it. And then weeks later, I get high fevers, I get swelling of my scrotum, huge infection, I'm septic, and I all I could do, I was so excited by the iron stuff. All I could do is think about maybe I am Drosophila. Maybe I can collect my urine and there will be hepcidin in it. And I did that. And that was the first milligram of hepcidin. So I started taking antibiotics and went to a urologist, was put on antibiotics, managed to stay out of the hospital, and collected a week's worth of urine from that septic episode. And that was my LPS equivalent and that was the first milligram because the hepcidin levels in urine went up 1,000-fold. So
Not
where I was
settled.
struggling to get the microgram, I got a milligram from myself.
That's amazing.
Yeah.
I love that story, Tom. I think I did.
Yeah.
I must have heard it, but I think I didn't remember it. It's
Okay. Well,
yours.
that's what happened. And then after that, our whole lab was collecting urine. There was a large jar in the bathroom, and we were isolating hepcidin from gallons of urine. But Nobody produced as much as I did. I'm still the champion.
I guess it's a good thing that you didn't get put in the hospital.
Yeah, it is. But I would have asked them to give me my urine back.
I think my daughters, when they had my grandchildren, I asked them for placentas and they were so offended. But I'm like, but I want to figure out how ferroportin and hepcidin work on the placenta. And they just kept saying, Mom, this is too weird. You can't have our placentas. I'm like, why do you care?
Exactly. So our graduate students delivered their placentas and they lived in the freezer and um and we had imaged ferroportin and a transferrin receptor on the syncytial trophoblast, some of the most beautiful pictures I've ever seen.
Really beautiful.
Yeah,
I know that work and the imaging was just
yeah,
great.
yeah.
Okay, Cade, let's get a couple more questions in.
Yeah, that was fantastic. You can't make stories like that up, which is just really, really fun to hear about. So yeah, you answered quite a few of kind of the questions we had through all that. So that was great. I did just want to ask if there were a few influential mentors or colleagues or trainees that you just wanted to shout out.
Well, I learned a lot from Diane and Jerry Kaplan. I learned a lot from Nancy Andrews. Um, but I have to say there's one other person I should mention who I actually never met, but who had a view of the iron metabolism systemic regulation that preceded hepcidin that just made things crystal clear to me, and that was Clement Finch.
Yes.
So Clement Finch, um, it was before my time, worked in the 1950s and 60s and 70s, but I had read his papers, and his view of iron metabolism essentially was the correct view, as it turned out later after all the research. He didn't know any of the molecules, or very few of them, but he had an intuitive sense of how iron was regulated, which was just unmatched. The way he explained things, he missed a few things, just to be completely accurate, but but what he described is what instructed my thinking, because I looked at his writing, I looked what he wrote, and I said to myself, it must be like that. I'm going to work on showing what the molecules are, but he's right, because he was what he was able to do is to do animal physiological experiments on iron in parallel with human disease studies. So, as an example, what he noticed is that patients who had iron deficiency anemia or had bled increased their intestinal iron absorption. But patients who had aplastic anemia did not, even though they had very high EPO, same hemoglobin, you know. So it made a difference whether you had hemoglobin of 8 and no active bone marrow, or whether you had hemoglobin of 8 and you had just bled out that amount of blood, or you were iron deficient. So your ability to absorb iron had something to do with the activity of the marrow. And that connection was cut and dry in an article that he wrote in the 1980s. And I said to myself, it must be like that. And one day I'll get a chance to work on that when we have the tools. And that turned out to be erythroferrone. So the work on erythroferrone was pre-imagined by Clem Finch. So he was definitely one of my heroes. I learned a lot from everybody in the field, uh, but but the way I think about this stuff is very much conditioned by the writings of Clement Finch.
I love that. I love that. The Cade, the community, the iron community through the years, and especially at all of these important discoveries, has been.
I think we lost your sound.
Oh,
Yeah,
you
I can't
lost.
hear you either. Oh yeah, I can hear you now.
Oh, I'm so sorry. Okay, sorry about that. I'll
The
move
iron
forward.
community.
Yeah, I was saying the iron community during that time was so collegial. Everybody just called and talked to each other and shared tools, and it, became close friends in these important discoveries. And it was beautiful to watch. It was really beautiful to participate and watch that sort of lead go forward in guiding the research of people.
And that is absolutely correct. And one other thing that I think is important in understanding, it was a very small community.
Yes.
You know, my mother would, who was still alive at that time, would say something. She said, how are you doing? What are you? Do people know who you are?" And I said, "Yeah, there are about 30 of them." And I'm very famous among those 30 people. And we are all very famous because we are in a small club.
Yeah.
So
That is true. It was a very small group. Everybody knew each other. And then when they started discovering all the ferroport mutations, everybody was in humans, everybody was sharing that information to try and understand that iron regulation piece in human disease. It was the way you hope most communities that are focused on a goal can work.
and it makes me, you mentioned ferroport, and we still don't know why we don't see those loss of function mutations that we are expecting. That would be there are missense mutations. Why are we that are essentially premature terminations or something like that, knockouts, complete knockouts of a copy of ferroportin? What we see is just missense mutations, substitutions,
Yeah.
and we are not seeing the mutations that completely inactivate the function.
Yeah.
Why is that?
It's a very good question because it's not, yeah. Well,
And it was an early question. We asked the question 15 years ago or more. We don't know the answer.
and
We
now
are
you know the
not
burning
seeing the
question.
expected frequency of mutations. We had all kinds of theories about multimerization and dominant negatives and this and that, but we don't have an explanation.
Yes. Still not.
Still don't have an explanation.
Yeah, and I'm not, I mean it flippantly, but does the NIH care? Would they fund figuring that
No,
out
no,
anymore?
this is inside baseball.
Yes.
This, yeah, this is something that troubles me, and troubles Diane, but doesn't trouble too many other people.
So it's hard to compel them that they think they know all that is told in this story, and we don't.
And there is this obvious kind of unexplained fact. It's not going to cure anybody if we figure it out, but it would make me happy.
Yeah.
Yeah, that's me coming into the iron field, in the last few years and reading all these foundational papers, it's so fun for me to hear about these foundational discoveries and where the field was at when they were made. I think sometimes it's hard to remember what it would be like to not know something, but just hearing the conversation here is just really amazing for me, to hear about all the amazing contributions you made to the iron field. I guess I also would just love to ask you, for people like me, younger trainees and scientists who are really interested in aspects of iron metabolism and homeostasis, what advice would you give to us?
Um, what I learned from this is that some of the rabbit holes that you get into turn out to be interesting. And this started out as a rabbit hole. And because I had a peptide that I had no clue what it was doing. And in fact, I said to myself at that time, it is a rabbit hole. What I can get lost studying this peptide because I don't have anything to go by. And and but sometimes the rabbit hole is interesting. So I think my advice to young people would be watch out for these things that are unexpected, and see if you can make sense out of them, because sometimes chasing the things that are expected is um, can get you papers, but it will not make you happy. I mean, it didn't make me happy to write papers on stuff that is expected.
Yeah, and the passion piece that makes you stay in
Mm-hmm.
are those little bit of rabbit holes where you find something that you're just um, surprised by, and that it just you get obsessed thinking
Yeah,
about it because it's so fun to think about.
yeah. Obsessed is right. And um, you know, let's be honest though, you have to also be a little bit lucky to come in at the stage of a field when something is totally unknown.
Yes.
And so I have to admit and that I was lucky. And my grandmother used to tell me, it's better to be lucky than smart. And I said, yeah. And uh, so I have to say I was lucky. But I think that if people are in a field for a while, you become lucky. You if you work at things, some things emerge that are new and interesting, and then you follow them.
Right. And you get a little bit of intuition as you're going along, like you talked about for Clement Finch,
Yep,
intuition about how this might work. And then people in the future were able to test it.
yep.
Yeah. That was,
And he was very, I would say, generous in describing ideas that he had about how things worked. We don't do that very much. We tend to hold things close now.
yeah.
But he essentially laid out for us in a few of his articles. This is how I think things work, and this is the evidence I have. Admittedly, it's not conclusive, and I don't know what the molecules are, but this is how things work.
Yeah. And I agree. I think, some of those pioneering articles by I would say hematologists at the time, and
Hmm.
and their seminal early thoughts really guided where we went because they were already formulating hypotheses that we had developed tools we could test.
Yep,
Yeah, yeah, it
very
was
much so.
it,
So
just
if
a
you ask me what the greatest influence in my life was, it will be him, Clem Finch.
I love that. I
Yeah,
really that's um, a great choice, Tom. I think that that's
he
a very
he made the
thoughtful choice.
choice.
Yeah,
Yeah.
yeah. Okay. Any other questions that you wanted to ask Cade at this time? How about the
Um,
big question in the iron field? Did we ask that? I know we had the ferroportin discussion, but
yeah. So I guess that's it. That's a good
And
one.
that's a little question that annoys me because I don't have a clue how to answer it. But I'll tell you, to me, what I would want to know. I would want to know the details of iron regulation, of how hepcidin transcription is regulated. I know we know quite a bit about it. We know it's centered on the BMP receptor. We know there are some accessory molecules but I want to know how the sinusoidal endothelial cell knows how much iron there is in the liver.
how
This,
it's sensing it.
I'm asking the question crudely.
Yeah.
I could say it nicer, but that is the question. The sinusoidal endothelial cell signals the amount of iron in the liver by secreting
Yeah.
BMP2 and BMP6. And I would like to know, how does the sinusoidal endothelial cell know about how much iron there is in the liver?
Right. Right. That's
What
a
are the sensors? How is the information? I mean, I know what it results in eventually in BMP2 and 6 secretion.
yeah.
But so when you look at any endocrine system, you got sensors, you got signaling pathways, and you got some kind of a downstream regulation of a target like hepcidin. So hepcidin is responsive to iron. I want to know what is sensing the iron and in which cells and how that information is distributed in the liver, so that the liver makes more hepcidin and there is more iron in the liver. I'd love to know that. I'd love to know the molecular details. I would like to actually be able to reconstitute that system in vitro, which we have never succeeded at doing. Put together endothelial cells and hepatocytes and make them regulate hepcidin.
Like
So
an organoid sort of thing.
yes, so you put in, we can do that now with inflammatory stuff like IL-6. We can put IL-6 on hepatocytes and they will upregulate hepcidin. But if we put iron on hepatocytes, it will do the hepatocytes do the wrong thing. They downregulate hepcidin.
Mm-hmm.
So you need the endothelial cell, which is a specialized endothelial cell, the sinusoid endothelial cell in the system, but we can't put it all together in a tissue culture dish and make it work. I'd like to see that work so that we can study it.
There you go, Cade. Your postdoc.
I have to keep that in mind.
When you finish your MD.
Yep.
That sounds good. Okay. This has been so fun. It's just been a great opportunity, Tom, to see you again, good friend, and to tell your history, the story.
Listen, we traveled together in this, and I remember what an exciting time we had and continue to have in some respects.
Yeah.
But how exciting those first steps were.
Yes.
And, what a wonderful opportunity it was. And you are right. What a wonderful supportive community the iron community was.
Yeah.
When it was small, when we could all get together. I'm trying to remember one of the early meetings we had.
Maybe in Italy or in.
No, there was one. I didn't make the one in Italy, but there was one in Bodega Bay.
Oh.
Bodega Bay was an early meeting, and we all fit into a relatively small room. It must have been an Iron Club meeting or something like that.
Probably.
Yeah, it was an Iron Club meeting, and it was early, and we all fit into a relatively small room and discussed iron. And it was just absolutely wonderful.
Yeah, really, really a great timing to be in that field of research.
Yep.
Well, Tom, thank you so much for taking the time to talk to us, tell us your history and story and the discoveries. There were so many areas we could have kind of gone down in the important discoveries that you and your lab and colleagues have made together. And I'm really glad to get this recording. It seems like every time I talk to somebody, it seems like we could have, 4 or 5 or 6 recordings of each individual. And we have, as you know, we have Nancy Andrews and Jerry Kaplan and Roland Lill, who are also going to still talk to us in the future. All of these recordings will be made available on the Center for Iron and Heme Disorders website, but also on our YouTube channel. So you can go and enjoy a history. And if we feel like
wonderful.
If we feel like this is a good thing, we'll go grab the next set of 5 people to interview and archive this history.
You didn't ask me about my trainees, so I think I'm going to do a closing thought, which is that none of this. We have a tendency to take credit for things we don't do, and so what I want to say is that the actual work done in the lab is what matters, and that work was always done by a team in my lab, and these people are the ones who actually created what I'm describing here, and there were many of them. But if I have to mention one person, it's Ella Nemeth, because she was the one who was there at the beginning, didn't know anything about iron, and responded to an ad, which was a spoof of the gut milk ad. And it says, gut iron, ? And it was gut milk was a campaign by the milk industry to promote the drinking milk. So there was a question, we made a spoof of that and pasted the whole building and several other buildings with this gut iron. We need somebody who at the postdoctoral level who is interested in pushing this iron field forward. We're at the very beginning. We described what specifically was going on, and she responded, and our collaboration is ongoing to this day. We finish each other's sentences, and uh, and she will, when I can't do this, she will still carry on. So that is one of the things that was a huge influence in my life, and many other people in the lab who uh, I should also mention Erica Valor, who and Chris Park, who purified hepcidin from urine, and many other people who I had the good luck to interact with. So, yeah, I thank you very much for the opportunity to tell the story. And yes, you're right. We could probably spend much more time doing it.
Well, and it seems like we need to. So we'll consider those opportunities, and I hope to get a chance to talk to Ella at some time too. So
All
this
right.
is great, Tom. Thank you so much, and Cade, thank you for your help in moderating this interview. And what I would recommend to you, Cade, is keep in touch with these people.
Yeah,
All right.
I'll let you. I feel like I could have asked 6 hours worth of questions, but it really was amazing hearing your stories and your perspective. And thanks for letting me be a part of this.
Thank you. Thanks for asking and
Okay, everybody, take care.
stimulating the questions.
Yep. Everybody take care, and I will send out an email when things get posted online so that you can share it.
Great. Thank
Okay,
you.
take care.
Bye-bye.